You're listening to WonderLabs, filling your earholes with nerdy goodness.
Hello and welcome to WonderLabs. I'm Chris Richardson.
And I'm Ellie Mackay.
In this episode, we speak with Rosie Davies. Rosie has a PhD in multi-omic technologies for rare diseases and is now working with a group at Great Ormond Street Hospital, aiming towards a personalised medicine approach within the rare diseases space.
In this conversation, we explore how multi-omic technologies have advanced since the days of the Human Genome Project, and we discuss potential issues around data security and the involvement of big pharma in academic research. We also explore one of Rosie's main research areas, a rare ciliopathy known as Bardet-Biedl syndrome.
Rosie and her group certainly have their work cut out as they compete for limited funding alongside several more common diseases, compounded by the emotional challenges of working alongside one of the world's leading children's hospitals.
This was a thought-provoking conversation about a little discussed topic, and we hope you enjoy finding out more.
Tell us a bit about your background and how you got into what you're doing at the moment.
First of all, I did my undergraduate at Imperial College. Then, when I was looking for things to do afterwards, I contacted this research group at UCL called CosGene. And essentially what they do is genome sequencing, looking for variants for mutations in patient DNA.
Mm-hmm. Hence CosGene as well, so that's Great Ormond Street.
Great Ormond Street, yeah.
Uh-huh. Okay.
Gene.
Great.
So they get patients from Great Ormond Street that are really difficult to diagnose. So clinicians have no idea what they have, or they might be treating them for one thing, but they're not entirely sure whether they're treating them properly because they've got no diagnosis for them. So then they sequence the DNA, send it to us, and we will analyse them, look through the genes, and try and find the mutation that may be causing the disease.
Okay.
So that's really investigative science, isn't it? It's
Yeah.
like you're the detective that's got to solve the...
Exactly,
solve the problem.
and that was really why I liked it, because I felt like I was like a detective. You do feel like you're a detective looking through someone's genome, and you're potentially the first person who's ever seen this person's genome there on a computer screen.
Wow.
It's really cool.
I suppose that brings us on then to the work you're doing at the moment. Your particular area of interest is multiomics. So
Yeah. What is that?
I mean, it can essentially be anything that you want it to be. I guess.
I love
that. Like, it's not a specific thing. It's multiomics is looking and studying multiple different types of data, I guess, is the best way to put it.
So the first one would have been genomics.
Yeah, genomics.
And that was the Human Genome Project.
Yeah.
Several years ago, we're just looking at the genome.
Yeah.
So now, this multiomics
Yeah, because there's
encompasses-
proteomics as well.
Proteomics.
And what's that?
Proteomics is studying the proteins of an organism. You have transcriptomics, which is looking at the RNA, so the transcripts that are transcribed before you start to translate into proteomics. So it's like a pathway between genomics, then transcriptomics, then proteomics.
So these are all the different stages from and you actually have the sequence of just letters, which is your DNA, the genomics, through to how those are translated turned into the proteins that are used to build various systems in our body.
Exactly.
Cool.
I mean, you can even go further. So you can go from proteomics to metabolomics, where you look at the metabolites that are then made. A protein will break down a substance and then you'll get a metabolite. But it can also be drugs that metabolize things, or food, or anything like that. It's the exposome, which I guess you could call exposomics, which is very catchy. That's not just what's inside you, but what you're exposed to in your outside life. So it's like your smoking habits, or
all the
how much you
environmental
drink,
influences,
where
beautiful
you live,
central London air.
exactly, and all of those things, and how they have an effect on your metabolite. Wow.
And that's a really hot topic because every single day in the news there is something causes cancer in our environment, and people want to know if they can influence their gene expression.
Yeah, exactly.
By eating a certain food or not eating a certain food, etc., etc.
And probably the answer is yes, though I want to say that a bit carefully. But
also just the sheer number of variables that feed into your personal risk for a lot of different conditions.
So that's why I say, multiomics is exactly what you want it to be, because, you can stick ohm at the end of anything that you want to measure and then it becomes an omic. So a lot more of these buzzwords are kind of coming out now.
So this whole body of information then really about what's going on inside the cell.
Yeah, exactly.
based on your own particular underlying DNA sequence.
Yeah.
Because that used to be an incredibly time-consuming, expensive process to actually sequence a genome. And, you know, we had the government racing against Craig Venter's private company, and now we've got these ads on the tube for these organisations where you can just send off a swab of some saliva and be presented with all this information about yourself.
So just to compare, the Human Genome Project, that was just for one person, took 10 years and cost over $1 billion. And now we've got 24 hours you can sequence.
Yeah, you could sequence probably less than 24 hours with new technology that's coming in.
It's amazing.
Yeah, it is amazing.
But multiomics then, I mean, it's really giving you so much more information. Do you find, you know, that it's meaningful and useful? How do you actually begin to sift through all of that noise to find the signal?
Yeah, nobody really knows, and this is actually what my PhD was. So multiomics is a buzzword really at the moment. Now we are into this what we call next generation sequencing era, which is essentially sequencing DNA and RNA. So now we're kind of in that era, people are thinking, right, well, now what can we do with this technology? And we've realised that perhaps the genome is not just enough. Like, you can find a mutation that causes a disease, but actually what does that do on a cellular level? You can't really answer that just through genomics alone. So that's why you need to add these extra omic levels. So the transcriptome will be able to tell you in a particular cell type, for example, say you've got a cancer cell. You know what the gene is, but what impact does that actually have on the cell? So you can then take the RNA from that cell and look at the gene expression profile. So you can see what other genes are being switched on as a result of this mutation.
Right. Oh, that makes a lot of sense.
Deep?
No, that's a really
absolutely
good explanation. I think a lot of people have the basic understanding that your genome is a sequence of letters and you can have a mutation in there. The normal metaphor we use is, you know, it's a book with sentences. But the idea is that the same spelling mistake in two different people may have a different outcome. And why is that? And it's because of this sort of pathway that you're describing, that a mutation at the genetic level, at the genome level, may have a different effect on the working of the RNA or the transcription factors or the metabolites or the enzymes. So that initial mutation can be presented or reflected in many different ways later on down the path.
Yeah, exactly. I mean, using that analogy, it's not only the single mutation in the DNA in that particular gene, but it's also all of the other mutations that you have as a background level that you've just acquired that by themselves don't actually make a difference, but in combination with this mutation that causes a disease, it can actually heighten or even suppress a symptom. So yeah, it's very quite complex. Yeah.
I think that's it. Because of this power that it has to answer so many questions, there's going to be a lot of unanswered questions at the moment. But given these next generation sequencing technologies, do you see the potential to twin that with other technologies like machine learning, artificial intelligence, something that's going to help you process and crunch that enormous volume of data?
Yeah, there are people that I work with who do machine learning.
I mean, presumably the increase in efficiency of the sequencing has happened because of and alongside the development of our computer technology and the ability for data to be crunched and processed at a faster rate.
As well as the storage of data and being able to comprehend the amount of data that's in a human genome. So if you put a whole human genome on a disk, it's about 1 terabyte of information. A long time ago, you wouldn't be able to fit that on a floppy disk or even a CD or a DVD.
Being several hundred CDs or something, wouldn't
exactly. But now we've got the capacity to be able to store thousands of genomes on one server. So technology really has had to evolve with where we want to be with sequencing. And
I suppose they both drive each other, don't they?
Yeah, exactly.
There's talk, isn't there, of using DNA as data storage.
Right. Yeah.
What's your opinion on that? Do you think we're going to get there?
So, I mean, at the moment we use a binary code, right, which is just ones and zeros. But DNA has 4 letters in the code, so you can essentially store more information on it, and therefore it might be much more informative. I think as a concept it's a very good idea, but the issue is being able to read it again. So you can store something like the whole of Shakespeare's collective novels or something on within some DNA, but then being able to go back and sequence it quick enough for you to actually take that information and be able to read it again may take quite a long time. As we said before, if it takes a day to sequence a human genome, then how long will it take to actually sequence that? It's not as quick as a computer yet.
So there's also some other concerns about just the sheer amount of information about people that exists out there. With multiomics, is that something that's also a concern?
I was actually having this conversation with a co-worker today. I wanted to really make sure that there there wasn't anything that as a negative point for getting your genome sequenced or even having it publicly available. And actually, fundamentally, if you just have a genome sequence, you cannot identify someone with that information. You could work out what their ethnicity is, you could work out what their eye colour is, you can work out certain things, but there's no way that you could actually get a facial profile of what they actually look like, or anything else like their age or where they live or any of that information. So to me, I don't actually think that there is any reason why people should be afraid of getting their genome sequenced, as long as they understand more of a mental risk of, do you actually want to know what's going on?
I think people have a lot of fear about big data, and there's a big thing of, I don't want insurance companies, I don't want them to know that I have this genetic risk of XYZ.
I'm sure that there's big data integrity policies in place at all the major institutions.
Hopefully.
Yeah. I mean, you did say actually about insurance companies, and I think that is something in the future that if everybody did get their genomes sequenced, if the policy surrounding it was not properly controlled, then that would be something that I imagine insurance companies would jump on.
Sure.
Like, for example, it's a thing at the moment, if you know that you've got Huntington's disease, you have to claim it. Inheritance. That is the reason why we do what we do. We can then do the research that we do at CosGene and pass that back to the parents, so they can then plan and do family planning and genetic planning. You know, is it safe to have more children, or what's the risk of having more children?
That's a really good point. Do you sequence the parents at all ever?
Yes.
You do?
Almost every time, because that's how genetics works, right? So in order to determine where the genes segregate, you can then work out which mutation came from which parent and how it works.
So you can blame which particular parent it came from.
Have you ever uncovered any particularly uncomfortable situations?
So there are occasions where you kind of look at it and you think, hang on a minute, this can't be right. Because you know, a mutation comes from a mother, and then you're trying to look for the mutation in the father, and you're like, hang on, this doesn't really weigh up.
That's some difficult conversations, isn't it?
Oh gosh,
what a way to find out.
Phrase as well.
Yeah, I mean, we do try and answer it. In that particular case, it wasn't the case that the father wasn't the real father, but actually that he had a very odd genetic abnormality, which meant that, and this might get a bit complicated, it meant that the particular gene that he meant to pass on didn't exist on one of his chromosomes, but it did on the other.
Okay.
And then in the offspring, they inherited the missing part of the gene from the father, and then the mutation from the mother. So that's...
Gosh, so it really is that, when you mentioned that detective investigative work.
Exactly.
It really is. I hadn't thought about that when you talk about sequencing children with these rare diseases. Of course, you're sequencing the parents as well, and you're looking back through the family tree and looking at the That's
very much what it is.
But in terms of clinical application of these sequencing techniques, using them to develop personalised medicine or different sort of technologies around diagnostics and things like that. I know that you worked on a particular disease called Bardet-Biedl syndrome.
Yeah, Bardet. So Bardet was French, Biedl German. So
international. Can you tell us a little bit about your work with that?
First of all, Bardet-Biedl syndrome affects 1 in 160,000 people.
Wow.
So there's actually probably around 500 patients in the UK who have this disease. And it's a ciliopathy, which is essentially a disease that affects the cilia of a cell. Quite a lot of people think they know what cilia are. As a biologist, when I first thought of cilia, I thought of beating cilia in the inside of a lung.
Yeah.
And they are a
Big
type
Mexican
of
wave.
cilia.
Yeah.
Yeah, exactly, yeah.
We're all in the studio, we're all doing a little Mexican wave with our arms here.
Yeah.
That is one type of cilia, so they're called motor cilia, which seems sensible when you are going like this. But the type that I look at are called primary cilia, which not many people know much about. And essentially, rather than having many of these cilia on a cell, there's only one, and they happen to be on almost all cells in the human body, apart from bone-derived cells like lymphocytes.
Oh.
These primary cilia, the main function of them is that they control sensory pathways. They also control a lot of different signalling pathways within the cell. So that is linked to Bardet-Biedl syndrome, because when you get a mutation in any of the Bardet-Biedl syndrome genes, of which there's about 22 genes, I think, that have been found at the moment.
And there's still many more potentially that we haven't found?
Potentially. So that 22 accounts for about 90% of all cases.
And do you mean you have to have a mutation in one of those, or all 22? Any one of those 22?
Any one of those. So it's an autosomal recessive disorder, which means that you have to have 2 mutations, so 1 on each chromosome.
Yeah.
So that means that you need to have 1 from your mum and 1 from your dad.
Yeah, exactly.
And that's what makes it rare.
The reason why it's so rare, they're quite important genes, so that if you get a mutation in those those genes, then it doesn't always mean viability.
So it's actually rare to have a surviving person. So actually you might
it's so fundamental, right?
It might be a very common mutation, but we never see it because those cells die off very early.
Yeah, exactly. A lot more money is being invested into investigating rare diseases. And this is very much reflected by GSK, who've just opened the Rare Diseases Lab.
Actually, I think, so this ties in with the landscape of the pharmaceutical industry. So what we've seen in previous decades are these so-called blockbuster drugs that tend to target diseases that affect millions of people worldwide. And the pharmaceutical companies have almost exhausted the list of diseases they can target on that kind of scale. So for their commercial approach, they're becoming more and more niche in the diseases that they actually target as well, which is great for these kind of conditions that have typically been overlooked. Pharmaceutical companies should make a return on their investment because that's what fuels funds research in the first place. And as we saw with the Human Genome Project, as we were talking about earlier, it was actually Craig Venter's Cellular Genomics that won the race and beat the public sector. You know, business can offer a lot to this field. It's more just many of these big diseases now aren't where they're turning their attention to.
Mm-hmm. How can you justify millions of pounds worth of funding? Playing devil's advocate here.
So, rare diseases, we work on them for a few reasons. One of which is, even though it's a rare disease, doesn't mean that the components of the rare disease don't affect lots of many other people. So, for example, in Bardet-Biedl syndrome, going back to that, one of the symptoms is blindness. And that affects many, many people. Age-degenerative macular degeneration. Is that the one?
Age-related
macular degeneration. That's the one.
That's the one.
That affects far more people. That's not a rare disease.
So that's losing your eyesight with age.
Yeah, exactly.
Seemingly niche, disconnected conditions actually have fundamental principles that we can use and apply to things that affect
Another one
people.
as well, Bardet-Biedl syndrome, one of the other symptoms is obesity.
Right.
Which of course affects huge numbers of people. And then of course you've got all the things that go with obesity, so you have got diabetes, cardiovascular research and loads of other things.
I think that's a very convincing argument.
It is also for scientific knowledge and widening our scientific understanding of how pathways work. And a lot of the time you may find a mutation in a gene that nobody knows what it does whatsoever. And then you can actually contribute something to the scientific community that you now understand what that gene does. So there's over 20,000 genes and we have a very minimal idea of what they all do. In terms of contributing to the science community, that is very fascinating, and there's a lot that we still don't understand about how anything works. And for example, a lot of the time you would have a patient that comes into Great Ormond Street and there are many clinicians who are working on this patient. So for example, you may have someone who's interested in the metabolics of how it's working, or there might be an immunologist or a cardiovascular clinician.
All the friends.
All of the friends. And each one of them are looking at this patient and thinking, oh, but this is my field, or this is my field, or this is my field. But actually, in reality, you don't know what process the gene is actually working in until you know what the gene actually is. Say, for example, you then find that gene and it's actually a metabolic gene and it's working in this pathway. Then you can then pass that information back to the clinician and they go, right, now we know what the gene is, we know how to treat it, rather than bombarding that patient with like tons of drugs.
Great.
And that happens countless times. An example of that is I have a friend who works on inflammatory bowel disease, and there's a very rare disease called very early onset inflammatory bowel disease, and they get incredibly horrible case of inflammatory bowel disease at a very young age, kind of younger than 1 or 2 or something. And in order to treat it, because it's an immune disease, you can treat it with bone marrow transplant.
Okay.
But in order to know that it's a rare genetic disease, you need to be able to sequence the DNA first. Otherwise, you could easily knock out the bone marrow. There's a 1 in 4 chance of survival, first of all. If you put a child through that when the outcome is going to be no different, then there's really no point doing it. And there's a lot of not only strain on the patients, but also the parents who have to make these decisions.
In terms of the work that you're doing at Great Ormond Street Hospital, which is internationally renowned as being a fantastic Centre for Paediatric Medicine, what's your experience been working at a place like Great Ormond Street?
It's hugely humbling. It is a very fascinating place to work because from a researcher point of view, you are working so closely with clinicians who then work with patients. So I don't actually necessarily work directly with patients myself. You get that kind of intimate perspective of seeing a clinician who works very closely with these patients and they know who they are. And
you don't have that much direct patient contact, but do you feel that the work that you're doing is having a direct direct impact on individual children? Do you get that sense of like, oh, today the work that I did saved this boy's life?
So we don't necessarily get that direct feedback, but there is a sense that what we're doing is definitely having an impact on them. And like I say, not only just the patients, but also the parents.
You're dealing at the sharp end of children's diseases. How do you stay positive?
Yeah, it's a very good question. I think because we are in a different building, I know I was saying, that it's very rewarding because we get to be on that end, but at the same time we are kind of separated because we are a research institution and the hospital is in a different building. We still have that connection, yet at the same time you have got that kind of way to kind of disconnect.
So I suppose that you sort of need to have that disconnect.
To survive it, yeah.
I guess that's the balance that everybody's sort of seeking, really, to do work that is meaningful but without destroying yourself in the process.
Yeah.
So, fantastic.
Exactly.
Great. Rosie, we've got a couple of rapid questions. If you weren't in your current industry or even working within science, what do you think you would have been doing instead?
I'd like to do music. I went with the science because I thought it was more stable, but actually now I'm there. I'm like, is it really?
I think a lot of scientists like music. I think it's something to do with the scientific brain tends to become a musical brain.
There's a hugely creative element to that work as well.
Yeah,
your lines of inquiry, you're developing hypotheses. And
it's the creativity behind it.
Which emerging field of technology are you most excited to be following at the moment and that you'll be looking at?
Probably genetics, but that's because I'm a bit nerdy about my field.
Anything to do with genetics?
It's not particularly current, but there was one a couple of years ago now where they treated with gene therapy for the first time, like a proper human. Really impressive, actually, that we've now actually got to that stage that we can do that.
That's something
gene
to watch.
therapy, definitely something to watch, and that's really kind of the next step after what I do is then trying to get to a point that we can actually cure people of these genetic diseases.
Is there a particular topic within your line of work or science more broadly that kind of frustrates you in that it's often misinterpreted or misunderstood or misrepresented by the media?
Probably nutrition. That just drives me crazy. Mostly because it's a fad thing, and I think everyone should just, eat healthily and
exercise,
balance. It's all about balance, right?
Do you remember a particular moment, at school or in your childhood, that you were first fascinated by science? The first moment you thought, this is for me?
I think it was all down to my dad, actually, because he used to be a science teacher, and I think he really captivated my interest in science.
Mm-hmm. Do you have a favourite piece of lab equipment?
Oh, wow. What a question. Yes. I can't believe I do, actually.
See? Everybody does.
Everybody's got their...
A multi-channel pipette has to be, the best piece of equipment.
Can you describe that for us? What is a multi-channel pipette?
So, a pipette, you can transfer liquids from one place to another. A multi-channel is multiplying a single pipette by, 8 or something.
I've seen them. They are cool.
You have, 8 tips, and it's brilliant if you need to...
It's an octopus pipette.
Yeah, it is an octopus pipette. So you just have your pipette,
I want one.
bung some tips on them, and then you can do, a whole 96 well plate,
Amazing.
So good. Yeah, all the way across.
Oh, that sounds exciting.
Yeah, it is very exciting. You don't have to do 90, because 96 well plates.
Yeah, try and do that individually.
Individually. Now you just have to do it 12.
Could you get a 96 channel pipette and just go poof?
They've made robots to do that now, but
Watch out, your jaw is going to be replaced.
I know, but
okay,
luckily
so
at the moment
everyone is going to be rushing out to buy a multi-channel pipette now.
Exactly.
Very cool. You could use that in cooking, couldn't you?
We'll
for icing or something.
Yeah, you couldn't do much more than a mil, if that's useful.
Very, very tiny doses of...
You could use it with an ice cube tray or something.
Or if you had 96 babies that all needed Calpol at the same time.
Yeah, exactly. But they'd have to be really close together.
Right, it's ridiculous. Okay.
Christopher, pull yourself together. Okay, finally, putting you on the spot, tell us your favourite science joke.
Right, there is a joke, but I don't think it's particularly funny, and I'm not sure if I can tell it well.
It's okay.
Go for it.
I'm going to try.
Yeah.
Okay, so there's 4 geneticists and 4 statisticians. They're on a train to Edinburgh from London, and the geneticists are like, we've got our tickets for the train. And the statisticians say, we don't need a ticket each, we only need 1 ticket. Right?
Okay.
And the geneticists are like, what? That's crazy. How are you ever going to survive with 1 ticket? Because there's 4 of you. And they're like, don't you worry, we've got a plan. So the ticket conductor comes into the carriage. They're like, tickets please, tickets please. The statisticians all get up and run out and lock themselves in the toilet. And then the ticket conductor comes down the carriage, takes the tickets from the geneticists, who are all fine because they've all got a ticket each, and then knocks on the door and notices that it's locked. So it's like, tickets please, knocking on the door. And they slip their one ticket onto the door to the conductor, and they're like,
sneaky.
Yeah, and they're like, oh yeah, thank you, and slide it back, stamp it, slide it back. And then the statisticians come out and go and sit back down with the geneticists. And they're like, oh my God, that was awesome. I can't believe we haven't thought of that before. It's really clever. So then, when they come back, on the way back to London from Edinburgh after their amazing conference that they obviously went to, the geneticists are like, oh, we followed your idea. We've only got one ticket this time. So it's going to be awesome. We're not going to have to pay or anything. And then the statisticians say, oh, but this time we don't need any tickets. And they're like, what? That doesn't even make any sense. So then as soon as the ticket conductor comes into the carriage, he's like, tickets please, tickets please. And then the geneticists run up, go to the toilet to try and do the sneaky trick that the statisticians did. But then the statisticians come out and knock on the door and they say, tickets please.
Oh, genius.
And then they slide the ticket over, steal the ticket, and then run straight into another toilet and take the ticket. Oh my God, that was a long joke. I'm really sorry.
I like that.
But anyway, there is a moral to the story, and the moral is that you shouldn't do what statisticians do unless you understand what the purpose of doing it is.
Right.
Yeah,
so there you go.
very clever.
And we've come away with some moral...
And the other
lessons
moral
as
of the
well.
story is always buy your train tickets.
Rosie, thank you so much for coming
Oh.
on, and keep up the good work.
Great.
Thank you
to
so
hear.
much. It was
Thank you very
actually, it was really nice just telling someone who cares about what I do. It's great.
That's the purpose of WonderLabs.
You're listening to WonderLabs, filling your ear holes with multi-omics.
Yay! Woo!